Great. So welcome everyone for the next hour. The whole field of biotechnology, including genetic engineering, is developing so rapidly, and we know that it has massive implications for nature, specifically the health and preservation of wild species, as well as for, of course, Indigenous rights and broader human well-being. And so with this really renewed government commitment and priority to halt and reverse the loss of species and nature, the way we manage genetic engineering of animals is a topic that we feel should really be on all conservationists' mind, all of those who love nature. Important time for it to be on people's radar. And while the technology is developing very rapidly, the field is still quite young, and we think we have the opportunity to influence its direction and to help protect what we value in the world by setting out some proper kind of regulatory parameters. It's certainly an important political opportunity in Canada this year for us to take action on this issue, because we know that a bill to amend the Canadian Environmental Protection Act will come before Parliament in the next few months. CEPA, as it's commonly known, regulates GE animals, and the bill is an opportunity then to put in place the safeguards for nature that are sorely lacking. I'm just going to ask everyone if they can to mute. I'm very excited. Some are sharing their morning coffee, but we can hear you. So if you're not on mute, that would be great if you could mute your microphone. Yeah, so we were just saying this is an important year because that bill, the CEPA legislation bill, is before Canadian Parliament. And so we thought it's an extra important time for us to kind of get up to speed. This is sort of an introductory webinar. We know we have people ranging from super experts to people that are just generally concerned about nature. So the plan for the hour is to sort of review the moment that we're at in Canada now. What are the emerging issues? Why has it become such a strong concern in Canada? And then secondly, to go look at the wider global context for how this issue is evolving. And we have two Marks on deck to walk us through the Canadian and global context. They're going to try to keep their combined presentations to about 30 minutes, and that should leave us about a good 20 minutes for questions and discussion. Let me just introduce our two speakers. We're quite lucky to have Dr. Mark Wells join us today from the UK. Mark, maybe you can wave and people can see where you are in the big box of squares. Mark is a biochemist with 9 years of research experience in molecular biology and biochemistry. He joined EcoNexus as a researcher in 2017, focused on a new form of genetic engineering known as gene drives. He's widely published in this area, including contributions to a major 2019 report on gene drivers, published by a number of highly regarded scientific networks, including the European Network of Scientists for Social and Environmental Responsibility. On this side of the Atlantic, we have Mark Butler. Maybe Mark, you can wave as well. He is a senior advisor to Nature Canada, working to ensure that the rapidly developing field of biotechnology does not have those unintended consequences for nature. Prior to that role, Mark was the policy director with Ecology Action Centre in Nova Scotia. So he has a good relationship with that shark in the bottom of your screen. Before that, Mark was a marine educator, a fisheries consultant, both in Canada and overseas, and has worked as a deckhand on commercial fishing boats. So he's been at this industry from all sides. We're lucky to have with us also as respondents for the Q&A section, Karen Wristen.
I think that they're, okay, so what you need from me is the start of a Google Doc, which is like,
Cassie,
theory of change, kind of like initial.
Yeah, there we go. Sorry about that. So just to say, Karen Wristen and Hugh Benevides are with us today to help answer questions. Karen is Executive Director of Living Ocean Society. You can give a wave, Karen. She's a lawyer familiar with CEPA, and along with EAC, that organization took the Government of Canada to court over the initial approval of GE salmon. Hugh Benevides is a well-known environmental lawyer with a long familiarity with CEPA. He has been advisor to many Canadian organizations over the years, and we're grateful to have his help with us on this file as well. So I'm going to turn it over to you, Mark Butler, to kick us off and provide some context and talk about Canada's experience, and then we'll go straight to Mark Wells after that. And keep your questions in mind, or you can start putting them in the chat, but we'll save them and come back at the end after both presentations.
Hi, everybody. Thank you so much, Gowrie, and thanks everybody for joining us today for an hour, particularly in this time of COVID and homeschooling, etc. As the title says, Nature Canada's interest in this topic is in what it means for nature and biodiversity. I'm going to provide some Canadian context, including about the world's first genetically engineered food animal, an Atlantic salmon, before we hand it over to Mark. And maybe I'm going to be hitting you over the head, but I just want to reiterate my main point at the start here, that I think this technology will have profound implications, both for human development and for nature. We are applying the tools of engineering to biology, as we did for chemistry and physics. And we're starting to see the results of this application of engineering, which is, putting things together, taking things apart, making things work. I just got my mRNA vaccine the other day. There was a news item on a genetically engineered pig heart for human transplant in the news a couple of days ago, and there's genetically engineered salmon, genetically engineered trees, etc. And I think these technologies will have major implications for nature, and let's get in front of it. Now, I realize, of course, that many of you are working on perhaps what you might call more immediate threats, from climate to habitat loss to environmental racism. And I have to say, I wouldn't be working on this issue if it hadn't popped up in my backyard, and if an American company hadn't selected Canada and Prince Edward Island specifically to develop the world's first genetically engineered food animal, the Atlantic salmon. And the reason I chose to get involved was because of the risk to wild Atlantic salmon, and also the fact that this is a first. And as environmentalists, we know, the value of precedent, and we know the value of social license. Now, I want to say this is probably a good moment for me to raise another extremely important issue with respect to genetic engineering, and that's the intersection with Indigenous people's rights. I live in Shabuktuk, or Halifax, on the unceded territory of the Mi'kmaq in Mi'kma'ki. And Atlantic salmon is a really important species for the Indigenous peoples of this region. They have a long relationship with this animal. And so that whenever this issue is discussed and there's a wild counterpart, the role of Indigenous rights needs to be part of it. And there's a great piece by Bob Chamberlain and Charlie Sark on this topic. I'll put a link in the chat later. And I think it'd be wonderful to see a webinar devoted solely to this topic. Okay, when most people, not you guys, but when most people think of salmon, they may think of a salmon fillet in the superstore, or they may think of the open net pen cages you see here, and then the pollution and impacts that come with them. But Atlantic salmon is it's a wild species. It's often called the king of fish. It spawns in fresh waters and matures in salt. The fact that it spawns in rivers makes the populations of those rivers genetically distinct from each other. And it also makes them vulnerable to genetic contamination or genetic introgression from GE or not, and non-GE fish. This is a map of the Atlantic salmon populations in North America. The colors don't matter, just to distinguish the various populations. But I think the important thing to remember from this map is that many of these populations are COSOIC listed. They're either endangered, threatened, or of special concern. So the species is in trouble. Whoops, I think I had a better slide than that. I did. There we go. So just recently, actually, and I was surprised by how many people, caught this bit of news and were kind of shocked by it. Just recently, in December, DFO and the Atlantic Salmon Federation were doing some monitoring on a river in Nova Scotia, and they detected a couple of escapees from open net pens in that river. And similar escapees have been discovered in other rivers in New Brunswick and Maine. And the point of this is that these domesticated, as the quote says, when domesticated salmon breed with wild fish, their offspring are less fit. It undermines, it reduces the health of the population and makes them more vulnerable to other threats also. And I was surprised to see that these escapees have ranged as far as Greenland. Okay, now, but we are talking about the risk from genetically engineered salmon. So the company AquaBounty has taken genetic material from two other species, a marine fish, an eel pout, and a West Coast salmon, a Chinook salmon, and inserted it into the genome of an Atlantic salmon, the purpose of which is for the fish to grow faster and to save the company money in bringing the fish to market. And I should, it's worth noting that under no circumstances would any of these species exchange genetic material in the wild. And this is just a cute picture of some eyed salmon eggs into which about 20 or 25 years ago, AquaBounty first injected this genetic material from the two other species. And it's this fish is a transgenic species containing material from other species. And this is a picture you see often when there's a story on genetically engineered salmon. It kind of drives me nuts because I think actually, after all this kind of hoopla about this species, I think there's good science to suggest that through conventional selective breeding methods, the salmon industry has now achieved growth rates similar to the growth rates that AquaBounty is claiming. So, apart from everything else, this fish may do nothing that we can't do with conventionally bred fish. This is a picture of AquaBounty's facility in PEI. They're now producing, or they say they're producing and putting fish onto the market. Of course, we don't have labeling in Canada, and AquaBounty is not labeling their product, so we don't know where it's being sold. As you see, it's on land, which definitely offers some containment and protection. Also, they are raising most of their fish are triploid, meaning they have 3 chromosomes, so they can't breed with wild fish. But there's documented cases of fish escaping from land-based hatcheries, and the triploid treatment is only 97, 98% effective. So if you have 1 million fish, which is easy in aquaculture, up to 20,000 of them could be fertile. And it only has to happen once for the wild stock to have the genes, for these genes to move into the wild stock, for these fish to reproduce with a wild salmon. Okay, DFO has done two risk assessments on this salmon, and I thought the science was quite good. Bob Devlin was the lead scientist for it. They did one in 2013 when they put some eggs in a cooler and shipped them to Panama, and they did one in 2019 on the facility you just saw. And this is the equation that they use. I think it's pretty standard for risk assessment. Hazard times exposure equals risk. In both cases, 2013 and 2019, scientists agreed that the hazard was high to wild salmon and to some other salmonids, but they ranked exposure as negligible in 2013 and is low in 2019. And so the end result, hazard times exposure equals risk, was that they ranked risk as low. And I would agree from that facility, although there's a salmon stream running through that facility, so to speak, or nearby, the risk is pretty low. However, what this ignores is the whole point of this is to grow the industry, to have many form of facilities, to be moving fish between the facilities, etc. So the risk assessment and the approval totally ignored the growth that you're going to get, the cumulative impacts. And actually there was a court case in the US that said that the FDA needs to go back and redo their assessment because they didn't look at the total impact. Okay, I haven't shown you, I don't have any slides of pictures of animals with two heads, but I think this is a genetically engineered rainbow trout, engineered to have more muscle and more flesh. This comes as close to a frankenfish as you might expect. And I'm just going to show you a few slides that show that this technology is not going to stop with salmon. There'll be West Coast species that are selected for enhancement, etc. All the phyla that we know may be considered for enhancement as this technology progresses. Just recently, Japan announced that they were approving two fish. Both of these fish have a pufferfish and a bream. Both of these have wild counterparts. I understand they're also looking at Pacific mackerel, and anybody who studies marine trophic systems knows how important fish like mackerel are in the system. And again, only has to happen once where one of these fertile fish escape and end up interacting with wild counterparts. And just on the left there in the left-hand column is the enhanced bream, and you see that it's slightly wider because it has more muscle and therefore a larger fillet. And let's not forget the hobbyists. Actually, the first genetically engineered animal to receive approval, I think in Canada and the US, was not a food animal, but was an aquarium fish. This is a fish that fluoresces because of properties that have been added. And I think, unless regulated, we'll see a lot more of this, fish, insects, perhaps reptiles, amphibians, plants. And in this case, a lot of these species have wild counterparts. Okay, just to repeat, top issue for Nature Canada is genetic pollution. When it comes to wild counterparts, there may also be competition for habitat, etc. And of course, the whole issue of Indigenous people's rights and appropriate engagement and the question of consent. Oops, sorry. There's lots of other, this is a big, sprawling, complicated topic. There's many other issues associated with it, patenting of life forms, and this whole approach tends to feed into a sort of corporate, large-scale, sort of designed food system, if you like. The whole issue of changing the genome, altering the inheritance of a species raises ethical and religious questions. There's definitely questions of animal welfare and also health concerns as you change the properties of an animal. So why now? Well, nature's in crisis. We just saw an example with the Atlantic salmon, and nature can't really absorb many more impacts or stresses. And many countries, including the G7, have committed to being nature positive. So, I think we should look through this, government should look at this technology through that lens and just the level of scrutiny should be so much higher. And of course, this technology is still very new, and it has both intended and unintended consequences for nature. And Mark can say more about that. And my point is, do we always want to be the people that show up in the ambulance, or do we want to try to put some barriers around this technology and protect nature in advance? And there is an opportunity to do that with the Canadian Environmental Protection Act, which a version of it was introduced in April of last year. Unfortunately, it contained relatively little about Part 6, so we're hoping when it's reintroduced in a few months or a month or two, that section will be beefed up, because maybe I didn't mention CEPA, the Canadian Environmental Protection Act, Part 6 of it regulates genetically engineered animals. So that's one place where the government of Canada can put in some protections for nature and respect for Indigenous rights. And I'll stop there and turn it over to Mark Wells. Thank you.
Go for it, Mark. Hopefully we can leave at least 15 minutes, or we can do 20 minutes or so at most.
Okay, yeah, thanks for the introduction, Gary, and yeah, thanks to Mark. Just trying to find my presentation. One moment. Sorry about this. It's there. Okay. So, yeah, just to introduce myself again, I am a researcher working for the organisation EcoNexus. We're based in Oxford in the UK, and we're a public interest research organisation. So my particular speciality is monitoring developments in a new form of genetic engineering called gene drive. But today I'm going to talk more broadly about where we are with genetic engineering and the impact we may well start to see on the natural world. And this is a vast field, so I'm not able to go into detail on everything. I'm not able really even to identify all of the risks in every area, but I will do my best to give a sense of the bigger picture and to pose some questions that I think we should be asking. So I'm going to talk about, well, first of all, what are GMOs? We'll have some definitions. And I'm going to look at the expanding scope of genetic technology over time. So we'll start with GMOs in food production. We'll then look at intentional releases of GMOs into the natural environment. And then finally, I'll look at what may well be the next step in genetic interventions in the natural world, which would be the release of gene drive organisms. And the case I'm making overall is that over time, we're seeing ever deeper and broader genetic intervention, genetic interference, if you like, in the natural world. So first of all, genetically modified organisms. So these are living organisms that have been modified in laboratories to alter their characteristics. Now, traditionally, the process of genetic engineering was done by introducing genetic material from other species. In more recent years, we've seen new techniques emerge that involve modification of the genome of a species without introducing new genetic material. So these techniques often involve something called CRISPR, which I imagine many of you have heard about. And because CRISPR has so much been in the news and the media, I think it's worth saying something briefly about CRISPR. So CRISPR is a new DNA cutting tool. It originates from bacteria, and it's essentially a programmable DNA cutter, and this can be used to disrupt a particular gene in the target organism, can be used to introduce short sections of DNA into the cut to alter the genetic code at a particular site, at a particular gene. And it can also be used to introduce large sections of DNA into the cut, often transgenes. And so it's a very useful tool. CRISPR has found all sorts of applications in basic research. But I'm going to be focusing on where we are seeing it applied in modified organisms that may be released into the wild. So you've probably heard of gene-edited crops. So these are crops that have been modified using CRISPR to disrupt particular genes without introducing transgenes. So there's 2 points I want to make with regard to gene-edited crops. So first of all, we still can't predict all the effects of the modification process, and so they still require risk assessing. Secondly, I don't think that transgenic crops, as in crops containing genes from other species, are going anywhere soon. Most of the traits that biotechnology companies want are very unlikely to be able to be produced by the gene editing process. They will require transgenes to generate herbicide tolerance or whatever. CRISPR is also being used now to produce the gene drive organisms that I will come to later in the talk. So its applications are very broad and are not limited to so-called gene editing. So I'm first going to look at the role of GMOs in food production. So very briefly to look at GM crops. The most common traits that we see in GM crops are broad spectrum herbicide resistance. That's by far the most common trait that is engineered in. And also in many cases, insect resistance as well by introducing Bt toxin genes from bacteria. So there's 5 GM crops currently commercially grown in Canada: corn, canola, soy, sugar beet, and a small amount of alfalfa. And my questions with regard to GM crops are, well, first of all, how well can agricultural methods based on GM technologies coexist with other methods, with conventional methods, with organic methods, where there is potential for crossbreeding of the different cultivars? My second question would be about resilience. So how resilient would food systems be if they are only based on a very small number of species, or strains even, sorry. And what will be the long-term effects on soil, insects, and other wildlife of these systems based on GM crops that also build in the use of broad-spectrum herbicides, often multiple broad-spectrum herbicides? So the use of GMOs in food production is obviously not limited to crops. We've just heard about the GM salmon from Mark, and this is interesting because it's the first commercially grown GM food animal, but as Mark's already told us, it's likely to be the first of many. There's a number of other food animals in the pipeline. But I'm going to move now from talking about GMOs in food production to what I see as the next level of intervention in the natural world, which is the deliberate release of GM organisms into natural ecosystems and into the wild. So in the case of food animals or crops, we may see sort of accidental escape of food animals, or we may see crops unintentionally growing as feral crops, but this is not the intention behind these organisms. So first example I want to look at of plans to deliberately release GM species into wild would be the GM chestnut. So the GM chestnut has been developed to be resistant to chestnut blight, and the intention is that it may, in some senses create a substitute for the chestnut tree that was largely wiped out in the wild by this blight. This is the intention. Whether it will succeed in doing that is another question. So approval is currently being sought to plant the GM chestnut in forests in the US, and it's likely to be sought after that in Canada as well. And this would be the first modified forest tree to be planted in North America. It would set a precedent, which may well lead to planting of other GM trees that are designed to improve their properties for forestry. So my questions here are, well, is the GM chestnut the same chestnut that it is intended to replace? Contains transgenes from a number of species. How will this affect its properties in the wild? What will be the effects if it interbreeds, or when it interbreeds, with the remaining wild trees? And why is a GM chestnut tree being brought forward when conventional breeding can also produce blight-resistant trees? So to move on to my second example of plans to intentionally release GM organisms, we're going to look at GM insects. So British company called Oxitec has produced a number of different modified insect species, and these all have the underlying aim of suppressing their wild cousins, if you like, either to suppress disease vectors like mosquitoes, or to suppress certain crop pests. So the idea is that the genetically modified males will mate with wild females after the GM insects have been released, and that in some iterations of this technology, this next generation, no viable offspring will be produced. Some iterations, no viable female offspring will be produced. So according to the developers, this technology should be self-limiting, that in theory, at least over time, we will see these genetic modifications disappear from the wild population. Whether this is what actually happens or not remains an open question. So already in the US, we've seen releases of Oxitec's GM mosquitoes, Aedes aegypti in Florida, and we've seen releases of GM forms of the pink bollworm and the diamondback moth, which are both crop pests. And the aim in all these cases is to suppress these wild species. To my knowledge, there's no releases of GM insects carried out yet in Canada. So my questions here with regard to the crop pests would be, well, how do the underlying problems come about? Why are particular crop pests abundant? What are the roles of our agricultural systems in creating the conditions where certain species become so prevalent? Secondly, what would be the consequences if the so-called self-limiting systems fail and these modifications spread into the wild populations of these species? And do we know enough to predict the consequences of such an event if we end up with GM insects living in the wild? Insects are much less well understood than crops. Do we know enough to predict what the knock-on effect would be? So I've covered the territory of GM food and intentional releases of what one might call conventional GM species, which are either taking place or in the pipeline. I want to move now to what may well be the next level of genetic intervention, which would be the release of gene drive organisms into the natural world. So gene drive organisms are a genetically modified organism like the others, but they have a very important new property. So the modified genetic material they carry, which is sometimes referred to as a gene drive elements even, is designed to override the normal rules of inheritance. So what would happen if gene drive organisms breed with their their unmodified counterparts, is that the selected traits that they're modified to carry would be passed on at a much higher rate than would normally occur. And they can be engineered to cause this higher rate of inheritance, even if the traits they're carrying are harmful to the organism. So, for example, if they're carrying a trait of female sterility or altered sex ratios. Very, very often they rely on CRISPR to achieve this altered inheritance. So the aim behind this technology is to rapidly alter the genetic makeup of wild populations. In some cases, to modify certain characteristics of the wild population. For example, there are proposals to modify, say, mosquitoes to be less effective disease vectors, perhaps. But more commonly, proposals relate to collapsing numbers of a particular species, or perhaps even eradicating them by spreading genes that interfere with reproduction in some way, often related to causing female sterility. So the term has been coined in French to describe the use of gene-derived organisms, forçage génétique, which translates as genetic forcing. I think this is a very apt description. And what we're talking about here is the proposed forced modification of wild populations. So if the GM chestnut tree is ever released, it may breed with wild relatives, it probably would. And in that case, we'd see the modified or the modifications it carries passed on according to normal Mendelian rules of inheritance. In the case of gene-drive organisms, something very different would happen, and I'll illustrate this here. So if an organism carrying a genetically engineered trait breeds with a wild relative, if that trait passed on according to normal rules of inheritance, we'd see the trait in 50% of the offspring. And again, in 50% of the offspring in the next generation as well. And so if there's no selective pressure, if it doesn't cause higher or lower fitness relative to the wild form, over time, the mutant or the modified form will not increase or decrease in prevalence in the breeding population as a whole. In the case of releases of a gene-drive organism, the gene-drive modified material will be passed on nearly 100% of the time. So in the next generation, all of the offspring from the mating event would carry the gene-drive material, and in the next generation too, and the next, and so on. And so what we see is over a period of time, which could be quite a short period of time in rapidly reproducing organisms, the modification could spread, or would spread throughout a entire breeding population. So no gene-drive organisms have been released as yet, but what we're seeing is the number of proposals to release gene-drive organisms increasing rapidly over time. So in some cases, the technology is quite advanced. In some cases, it's less advanced. So what are the proposed targets? Well, mosquitoes are what you might sort of call the highest priority target, where the technology is most developed, and we may well see releases of gene-drive mosquitoes in the...
Mark, can I just ask you to try and wrap up in about 2 minutes?
Yes, I think I'm on course to wrap up in 2 minutes. Yeah. The technology is also likely to be applied to agricultural pests, the suppression of these pests, possibly also to modify them to stop them spreading pathogens. And it may well also be applied to invasive species. You may have heard of proposals to suppress invasive mouse populations on islands by releasing gene-drive mice. But I think what I note here is that there's no evidence yet that the technology can work in mammals. And so I am concerned that it may be oversold with regard to its possible applications in conservation. So I just briefly want to look at the risks and uncertainties with regard to gene-drive organisms. So even if these behave as planned, there will be ecological impacts. But we also face risks of, for example, uncontrolled spread of the gene drive genetic material. We face risks of gene drive elements moving into other species. The effects of gene drives at a genetic level are very likely to be irreversible. Many of the gene drive designs involve inserting CRISPR permanently into the genomes of wild species. And so regardless of if the gene drives work or not, we may well see CRISPR genes in the wild with unknown consequences. And given the complexity of the systems involved, I think there's clear potential for effects we haven't predicted. And any of these outcomes could lead to further ecological effects. So just to summarize, what we're seeing is an expansion of the scope of GM technology from use in food production to deliberate release of GM organisms into the wild, to perhaps in the next decade, the deliberate release of gene-drive organisms into the wild that will force modifications into wild species. And so I think we're looking at an issue here that isn't going to go away. I think that as the technology advances, the likelihood of harmful impacts increases as the number of events of escape of transgenes or deliberate release of transgenes increases, the more the likelihood of a harmful outcome or multiple harmful outcomes increases. So I think this is something that may well unfold over decades, but it's certainly an issue that we need to, I think, very proactively engage with. And I will leave it there.
Well, thank you very much to both Marks for very comprehensive and stimulating overviews of the challenges generally already with salmon and GMO food crops in Canada, GMO food animals, and the potential direction that the biotechnology firms are going in. It raises a lot of important questions. So I am just looking for you, if you, on the bottom of your screen, you have a reactions button, which allows you to raise your hand if you want to ask a question, or you could drop one in the chat. You can ask a question to either Mark, and we also have Hugh and Karen on hand if you have some very specific CEPA questions. There is an initial question, which you can probably see. Can you speak briefly or suggest other resources on the risks of GE trees in nature? It seems the Forest Stewardship Council is discussing removing the prohibition on GMOs. It would be good to get a sense of how much of a threat these are, because of course, she's pointing out that FSC is looked to as a gold standard. So does either Mark want to comment on the risk of GE trees to forest health overall and what that should mean for the Forest Stewardship Council?
I'm afraid that's a bit beyond my speciality, but I can see we do have Lucy Sharrett with us who may be able to say something to that.
Mark, Butler, if you come off mute, then I'll know you want to answer. And oh, you are, okay.
No, I don't want to answer.
Oh. Lucy, do you want to speak to that?
I don't really have an answer to the question other than the question of risk assessment has been discussed, but not rigorously, and I can put a resource in the chat about it. There's just so many unknowns And yeah, so the question is, can you actually do a risk assessment for GE trees before you release them into the wild?
Presumably the general concern about risk is there. It's just a question of how you kind of specify it, because the point is that the trees can interact with other parts of the forest. Great. Well, we'll look forward to, if you're able to dump a resource in the chat. There it is.
Maybe I'll just add, and this is not a scientific point, but more of a policy or political point, to say that, you know, the industry is looking for social license. You know, I think the industry, forest industry, presumably would love to bring, you know, launch plantations containing GE trees, I don't know, a GE eucalyptus or a GE spruce, but instead the first tree to be brought on the, you know, to be brought forward is a GE chestnut, which, you know, as a conservationist and a forest lover, I see the appeal. So I think, you know, to some extent, the industry, it's looking for social license and hoping that the GE chestnut will give them that.
Right, good point, because they're addressing a species where there already is some concern. Welcome other questions or also just comments. If you share the concern about how CEPA is or isn't addressing these issues, or other thoughts you've had that have been prompted by our speakers. Simon at EAC, go ahead.
Sure, thanks, Scoury, and thanks to Mark and Mark for the presentations. I guess the question here is kind of related to the difference in like value judgments on certain kinds of GE initiatives. Like obviously we share Mark's concern for you know, the case of wild Atlantic salmon, but then as was just kind of touched on, maybe there are some mixed feelings on, you know, a situation like the American chestnut. How does that look in your mind? Maybe this is for Mark Butler, in CEPA, when we get down to sort of shifting the legislation to accommodate, you know, two examples in that case that could be very different. What does that look like for you?
Good question, Simon, and I might pass it over to Karen and Hugh too to elaborate further. You know, we want to signal, at least in the case of the accidental movement of genetic material into wild species, or through the accidental release, that there needs to be extra protection, or, you know, this is pollution that doesn't just persist forever; it actually grows over time, and you know, as we know with invasive species, can't be pulled back. So we want the highest level of protection for our wild species when at a time they're already in trouble. And, you know, despite some of the good science that DFO did, in the end, I think their methodology failed, and we're now growing this fish right in the middle of the range of the Atlantic salmon. So, you know, we should treat, when there's a wild counterpart, which also raises questions of indigenous rights, which haven't not been addressed, you know, these should be treated as the worst of the worst chemicals also. And that is certainly not the way the government is treating them right now. I mean, there's more I could say, but maybe I'll stop. And if you or Karen want to add anything to that.
I would just say, just reviewing the current Part 6 of CEPA, which is the Canadian Environmental Protection Act, which is supposed to be the mechanism for assessing new organisms in effect. So Simon, looking at that opening part of Part 6, if you say you want to introduce a new living organism or what's called an animate product of biotechnology in significantly greater quantities or concentrations, or in ways where there'd be significantly different exposure, potential for exposure or actual exposure compared to now. Presumably any new organism would meet that criteria because it didn't exist before. Then you'd have to go through the process in CEPA. So my answer to that, we've been focused mostly on new animals that would affect a wild counterpart, but that concern could spread or could apply to plants as well. Our concerns are that, and arising from the court case that Karen and Mark participated in, was that you often don't know that some applicant wants to introduce that new species, or that they've been granted a waiver of providing certain information for the purpose of the risk assessment until perhaps well after that assessment is underway or completed. So I'm just trying to answer the question that we haven't seen the kinds of improvements where people, the public, could meaningfully participate or retain their own experts to participate in those risk assessments for any new organism, plant or animal. And that's the kind of meat and potatoes, basic public involvement and starting with public notice that we're looking for in CEPA that we're just not seeing.
Karen, did you have a comment you wanted to add?
No, thanks. I think Hugh's covered the only point that that I wanted to make. We don't have the points of access into this discussion that we should have.
So it kind of speaks back to your point, Simon, that if you had the opportunity to weigh in and assess individual cases, there's the possibility for different levels of answer for different kinds. When you're saying, how will the Act handle something on a continuum, potentially from a chestnut to a salmon, without assuming one is good and one isn't, but there is no opportunity to weigh in at all with other expert views, is I guess what I hear Hugh saying. So we need those points of deliberation. Hugh has a follow-up.
Very quickly, I just neglected to say that CEPA is designed as the residual legislation. That is to say, if the federal cabinet decides that another law, federal law, is up to the task of assessing and managing, quote unquote, a substance, then that other act would apply. So there are acts involving animal feeds, for example, and seeds, which take effect if that new organism is, for example, fits under those other laws. So that's always another concern that we don't. And then tomorrow, the cabinet can always add another law to that list of laws that applies instead of CEPA. So that's just a mechanism that I think is important to note is there.
Great. Inka.
It seems to me that the kind of wedge point, or I'm not sure what the language is, but to me that whole risk assessment process has been problematic for as long as I've been involved in sort of marine conservation issues, and not just marine, but terrestrial, food issues, pesticide issues. And, you know, asking the public to engage on a process that involves a risk assessment is really difficult. I mean, I just, you know, the analysis that's provided to the public around these risk assessments are sometimes very arcane mathematical models and a lot of verbiage that the public doesn't really understand. And so for them, for the public at large, they're only going to, you know, in some ways, well, I don't want it. And that kind of feedback to government isn't, you know, the government isn't receptive to. So I want, you know, I think this is an area we need to tackle. And Hugh, you mentioned that it's a part of the Act, but, you know, there's so much science that's been produced around risk assessment and how it should be done and what are the best ways to do it and what are, but this is not coming into play into policy, and that those policies aren't really implementing the best science practice around risk assessments. So it's just a comment. It's just something that has frustrated me for a long time. And the Canadian government has a history of doing risk assessments on various things that have, with time, I'm particularly thinking around metal contamination, pesticide contaminants, or pesticide products, metal, and which over time they've constantly ratcheted down the limit, the level at which those concentrations are deemed a human or an environmental health hazard. So I have very little faith in even if that is in the language to do a risk assessment, that risk assessment is going to be meaningful over time.
Hugh, do you want to just pop in for a few seconds on how we would handle the public doing assessments or not? Because then we will have to wrap up.
I agree with Inka, and I would perhaps there's an opportunity for Mark Butler and me to talk to you about these problems. I did purposely say a sort of an addendum that the public can bring its own expertise in, because in this area and others, there tends to be a monopoly of government science and, frankly, proponent science. And so I thank you for that comment. And the first thing that we're trying to do is ensure that there's greater notification or greater notice. And then you encounter all kinds of other problems, some of which you've identified. And it's lovely to see you after many years after our encounters involving ivermectin in aquaculture, I believe, if my memory serves.
And Gary, if I could just say real quick, the Canadian public has said no, or a substantial portion of them had said no, but government tends to dismiss that, I think, as kind of anti-scientific. And I think what we're trying to do is that there are scientific grounds to be concerned here. Thank you.
That's right. So the opportunity to intervene allows us to bring expert opinion to bear is part of the point. I want to thank Mark and Hugh and Karen for being with us as expert resources today on what is really a growing and important issue, and to all of you for joining us for this webinar. Stay tuned. We will continue to send out updates for ways you can learn and follow us, and there's a lot of important work to be done with reference to reforms to CEPA this year, so we'll count on your support as well for that. Thanks to all, and have a good afternoon.